Hello everyone, welcome to our new video. In previous video, we have talked about photoperiodism. What is photoperiodism? What are the type of plant on the basis of photoperiodism? And some experiment related to photoperiodism. Now we are continuing that series. In this video, first of all, we will study about site of perception of photoperiods. फोटोपीरियड्स कौन सा प्लांट का वो ऑर्गन है जो एक्ट करता है एज ए साइट फॉर रिसेप्शन ऑफ दैट फोटोपीरियड्स? एंड आंसर इज लीफ. प्लांट का लीफ पोर्शन है प्लांट का जो लाइट को रिसीव करता है और रिसीव करने के बाद उसको ट्रांसपोर्ट करता है टू transport that stimulus to shoot apical meristem or shoot apex where flowering will takes place. Selection performed some experiment that prove that leaf is the main organ for perception of photoperiods. They experimented on chrysanthemum and gentium plant. For example, this is a short day plant. This short day plant under short day conditions will flower. If there are leaf on the plant and we provide it short day conditions, it will flower. In second case, all the leaves are removed and no flowering will be seen in this case. In third case, a single leaf is present here and this leaf will receive photoperiods for short days. Because it is a short day plant and one leaf is present, in this case flowering will occur. This study shows that even one leaf is enough to receive all the photoperiods required for flowering. Now study the similar case under long day conditions. All three cases are studied under long day conditions. When we are providing long days photoperiod to this short day plant and leaves are present here but there will be no flowering even if lots of leaves are present here. In second case, we have again removed leaves and again no flowering. In third case, one leaf that is from short day conditions is grafted on plant which is under long day conditions and it will give flowering. Only a single leaf is under short day conditions and the whole plant is giving flowering. That means a single leaf is enough for a plant to give them flowering induction. Now understand some more grafting experiments. In first condition, this is a short day plant under short day conditions and we have taken a leaf from this plant and transferred it to non-induced plant that is not under short day conditions. Now we will put this second plant into long day conditions. But in this case we have one leaf from the short day conditions induced plant and due to presence of this leaf under long day conditions, plant will flower. So from all these experiments we can conclude that leaf is the main organ that is responsible for flowering, and even a single leaf is enough to carry these photoperiodic responses. Now let's see what is the transmissible signal that is required for flowering. For flowering, there is a special difference between site of perception, that is leaves, to site of action, that is shoot apical meristem. Shoot apex or shoot apical meristem means there must be some signal that is transferred from leaves to shoot apical meristem for occurrence of flowering. Mr. Chalakian first of all talked about florisgen hormone. Means florisgen is the substance that is responsible for flowering, or it is transported from leaves to shoot apical meristem, and this is the reason behind flowering. Mr. Chalakian performed some experiments, and in first experiment they collaborated some plants. For example, this is a plant stem of gentium, and they fused this plant to next gentium plant, and this gentium plant again fused with next gentium plant, and so on. This gentium is put under short day condition. Short day conditions. Since it is a short day plant and it is getting short day conditions, it will show flowering. But these plants are not under short day conditions. So some chemical substance will transferred from this gentium, which is taken under short day conditions, to this gentium and this gentium, and both of them will show flowering. There are some grafting experiments that are performed by Chalakian. In these experiments, they grafted Nicotiana with Hyoscyamus. Hyoscyamus is a long day plant and Nicotiana is a short day plant. There are three conditions applied to them. In first condition, they provided them short day conditions and both of them flowers, because Nicotiana is a short day plant and there are short day conditions. So this Nicotiana is responsible for flowering in both the plant. In long day conditions, both of them again flowers, because Hyoscyamus is a long day plant and it will give signals for both of them. In third condition, when we put Nicotiana without short day conditions and Hyoscyamus is a long day plant, there are this is in short day conditions. If we fused after that, then no flowering will be there. What we can conclude here, that there is some chemical present in these plants that is secreted on their particular conditions and it is transported to their shoot apical meristem for flowering. Phytochrome. What is phytochrome? Phytochrome is a bilirubin that exists in two forms, PR form and PFR form. this PR form get converted into PFR form after absorbing red light of 660 nanometer, while PFR form again get converted into PR form after absorbing far red light, that is of 730 nanometer wavelength. This phytochrome is the main photoreceptor for photoperiodism. This red light can inhibit the plant from flowering in short day plant. Let's see some experiment with this phytochrome. Here are some experiments related with short day plant and long day plant. These are the light periods given to the plants. So in first condition, light period is more than the critical dark period. So in this condition, long day plant will flower, while short day plant will remain vegetative. In second case, light day is short than critical dark period. In this case, short day plant will show flowering, while long day plant remain vegetative. In third condition, this is the light period that is shorter than dark period, and we will give it some flash of red light here. And in this case, long day plant will show flowering, while short day plant remain vegetative. So this red light flash is responsible for not producing flowering in short day plant. In next case, this red light band is followed by far red light band. Now this far red light will inhibit the effect of this red light. So in this case, this plant, long day plant, will remain vegetative, while short day plant will show some flowering. In next case, red light is followed by far red light, and again it is followed by red light. In this case, this results will be shown by plants. So long day plant will show some flowering, while short day plant will remain vegetative. In next case, red light followed by far red light, again red light, and again far red light. So this red light effect is inhibited by far red light or reversed by far red light. So in this case, this plant remain vegetative, while short day plant will show flowering. So in this experiment, we can see the role of phytochrome. This phytochrome is responsible for this red light, far red light effect. Now let's see vernalization. Vernalization is requirement of low temperature. and this temperature is given to plant to hasten flowering, to produce flowering earlier. For example, like winter plant, plant variety of wheat. If we provide it low temperature treatment, it can flower like spring wheat variety. Clip art. First of all, observe this phenomena of vernalization. But first scientist, that is Linsco provide evidence for vernalization. Linsco worked on wheat varieties he grew winter wheat in spring season, and this winter wheat failed to flower. Then he provided it low temperature treatment and this winter wheat can grow in spring season also. From here we got the term vernalization. For vernalization responses, there are different time periods for different plants, like for Hyoscyamus 10 days, Oenothera 30 days. For different plant there will be different periods. Site of vernalization. The effect of vernalization is perceived under shoot apical meristem or shoot apex. Devernalization. Process of this vernalization is reversible and can be cancelled, and this process is known as devernalization. How we can do devernalization process? By giving higher temperature after giving low temperature. Higher temperature instantly by drying vernalized seeds for long time, or by providing anaerobic conditions, because for germination of seeds oxygen is required, water is required, and we are removing both these required conditions, so we can stop vernalization, and this process is devernalization. Now let's see vernalization with gibberellins. Vernalin is the main hormone that is responsible for this vernalization process. This vernalin is present in long day plant and short day plant. In long day plant, vernalin get converted into gibberellins and anthocyanin is also present there. both anthisyn and gibberellin will give rise to flowering. While in short day plant, no anthisyn is present there and vernalin is not get converted into gibberellins. So, there will be no flowering. What are the practical applications of this vernalization process? Let's see some practical applications. First one is we can cut vegetative period and enhance flowering early. So, life cycle of our plant get short. So, it is a time saving process. It increases cold resistance in plant. Cold resistance in plant. Vernalization process can be transferred through grafting procedure. So, this feature can be used into horticulture. And we can use vernalization into crop improvement. Crop improvement. Now, let's discuss some MCQs on photoperiodism and vernalization. First question. Fill in the blanks influences the process of flowering in plants. Photoperiod, water in the soil, the acidity of the soil, D, amount of green pigment. So, it is the day length and night length that influences the process of flowering in plant. So, right answer is A. Next question. Who discovered photoperiodism? Darwin, Linscoe, Arnold, Gardner and Allard. Gardner and Allard were the scientists who discovered photoperiodism in tobacco variety. Next question. Which of the following plant is LDP? Tobacco, Glycinax, Mirabilis jalapa, spinach. And right answer is spinach. Next question. Photoperiodic stimulus is perceived by leaves, flower, birds or meristems. And right answer is A. We have seen many experiments that show that leaves are the organ which perceive photoperiodic light. Next question. Maryland mammoth is the variety of soybean, wheat, tobacco, rice. It is that variety on which Gardner and Allard worked. So, it is a tobacco variety. Next question. When the dark period of short plant is interrupted by brief exposure of light, then the plant produces more flower, will not bear any flower, turn into a long day plant, produces flower immediately. We know that dark period of short day plant is very important for it to flower. When it is interrupted, the short day plant will not bear any flower. So, right answer is B. Next question. Which of the following pigment involved in red, far red light conversion? Cytochrome, lycopene, phytochrome, xanthophyll. Right answer is phytochrome. Phytochrome is the pigment which can be converted into PR and PFR form. Next question. The wavelength absorbed by PR form of phytochrome is 640 nanometer, 660 nanometer, 720 nanometer, 740 nanometer. Right answer is B, 660 nanometer. Next question. Chalakyan named photoperiodism phytochrome, dormancy or fluorine. Chalakyan discovered fluorine hormone. So, right answer is D. Next question, the pre-showing low temperature treatment of seed is known as scarification, electrophoresis, vernalization or abscission. So it is known as vernalization. Right answer is C. That's all for today's video. Thank you so much.